Hello everyone, this is Dr. Fermani with the instructions on how to do lab number 11 online. So lab number 11 has to do with eukaryotic kingdoms. This coincides with chapters 15 to 17 in your textbook. I highly recommend going over the lecture materials on these chapters before beginning this lab. I also highly recommend reading the whole background and introduction to this lab, and also the introduction to the different parts of the lab before continuing on working on this lab. After you have done that, then I would come back and work on your pre-lab questions. So for your pre-lab questions, you have 12 of them. So just like before, when you go into your Word document, just make sure you have your name, what lab you're working on, and that they're your pre-lab questions. And just simply just write all your answers 1 through 11 on this page. Once you have finished your pre-lab questions, then you can move on to the protocol of the lab. Before I move on though, however, I just really want to point out this diagram. And this is really important so you understand the basis of this lab. So we have talked about the 3 domains of life: bacteria, archaea, and eukarya. Right now we're just focusing on one domain of life, eukarya. And within eukarya, it can be broken down into 4 kingdoms. So the 4 kingdoms are Protista, fungi, plantae, and animalia. And through this lab, you're going to investigate every single kingdom that you can find in eukarya, called eukaryotic kingdoms. So going on through the introduction, we're going to come down to part number 1, which is on the first kingdom, Kingdom Protista. So through this protocol, you would have done some observations of different kinds of protists. So what I just did is I found some videos and images to supplement what you would have done in class. So here, the first part is to analyze live specimens of Euglena and Paramecium. So what I did was found some videos. So here's Euglena. And what I really want you to just know is how the Euglena is moving, its locomotion. So I'm going to just play a little bit of this video and just take a minute to see how the Euglena are moving, what they're doing. Okay, and next are the Paramecium. So here are the Paramecium. I'm just going to play a little bit of a video. Just to let you know, I have no idea what this huge thing is. Pay attention to these smaller guys. Some of them will not be moving. They could have just gotten kind of sick and started dying while the slide was made. So I would really just pay attention to the live swimming Paramecium and how they're moving. So here you can see some of them. This guy's going to fly through. This guy's going to fly through. Okay. Okay, coming back to your protocol now. So we covered the procedures, parts 1, 2, 3, and 4. Part 5 is to look at some prepared slides. So you would have looked at the amoeba, volvix, and plasmodium. So if I come to my protist images, so here's an example of what you would have seen under the microscope. Here's the amoeba. You can see these little arms coming off called pseudopodia. Next is the volvix or algae, and you can see how crazy they kind of look. And then lastly is the plasmodium. So just from the plasmodium, what I want to tell you about it is you see these little red dots. And that is not the plasmodium. It's actually where the arrows are pointing to, those little purple specks. That's the plasmodium. That's the protist that you're supposed to be finding. And one of your post-lab questions have to do with these red dots. So think really carefully, what are all these red dots? Why would they be in this microscope slide? So in particular, on the bottom of page 125 is a paragraph about plasmodium, kind of like where it lives and where you can find it, and it will definitely give you the answer to what those red dots are. So the last thing that I just really want to point out is that these are all protists. They're different kinds of protists, but I just really want you to see how diverse protists are between each other, and that really shows between these 3 different microscope images. Okay, so coming back to your protocol, that finishes part 1 on protists, and now we go to part 2, which is on kingdom fungi, so fungus. So the first thing you would have done is look at a prepared slide of mold types. So if we come to here, so here is an example of different types of mold under the microscope. You can clearly see how different they look from each other, and this is just observational, and you have a post-lab question on this. Coming back to your protocol, after you looked at the mold types, then you would look at live cultures of Penicillium and Rhizopus. So we come back to fungi. So here is penicillin. This is grown on a petri dish, okay? And here you can see a nice culture of penicillin. If you were to zoom in a little bit more, you would see these defined structures. And here's Rhizopus, which looks a little bit different, you can tell. So here again is penicillin, okay? And here is Rhizopus. So these are, again, observational. You have a post-lab question on them. Next in your protocol would just be to observe a spore print that was made from the mushroom cap, putting it on a piece of paper. And you can see the spore outline, so I will show you that. And the last part is just to go over a little bit of the anatomy of a white mushroom. So here, if I go to the fungus, so this is a spore print. So you would see all the spores that went onto the paper if you pushed the cap of the mushroom onto the paper. And here's the anatomy of a mushroom. Here's a little bit more of a detailed diagram showing you the parts of the mushroom. And here's white mushrooms. You've probably seen them before. You cut them in half. What you can really see is the gills right here that contain the spores. And you can identify the other parts of the mushroom. And this is just, again, observational. Okay, so that was part number 2. Part number 3 was on Kingdom Plantae, or plants. So here is really important to understand how plants evolved. So first you have the bryophytes, which are moss. Then you have ferns. Then you have the gymnosperms, which are pine trees. And then you have angiosperms, which are flowering plants. So we're going to look at how the plants have evolved from moss all the way to flowering plants. So for each section of the plant, you would have looked at a live specimen and then a prepared slide of the life cycle. So when I go to the images, we're going to look at that for bryophyte, ferns, and gymnosperms. And then when we get to angiosperms, you would actually had a flower. You would identify the anatomy of the flower, and then you would have done a small dissection by cutting the ovary of the flower in half and seeing what was inside. Okay, so here I'm going to come to my plant images. So this is what you would have looked at. So we would have had a live specimen of moss in the lab, and I would point out 2 different things, and this is really important. There's 2 main structures that I want you to know, the gameophyte and the sporophyte. So I'm going to come really quickly back to your lab, and I just want to really point out this diagram that was in the introduction here. So this is called the alteration of generations, and it shows the 2 main forms that the plant is in. It can be in a gameophyte or a sporophyte. So the gameophyte is haploid, okay? It will reproduce these cells through mitosis. So we know mitosis is just a copying machine. So it's just going to make lots of copies of the gameophyte, okay? The gameophyte is important for making gametes, which we know are eggs and sperm. So it's going to make a ton of these gametes, and then these gametes will come together for fertilization. So if each gamete is haploid, or N, when you put 2 of them together, sperm and an egg, they become 2N, a zygote. And now that zygote will start dividing through mitosis, so it will stay diploid and make the sporophyte. So this structure, the sporophyte, is important for making spores. However, it starts off diploid and it ends haploid. So what process does that have to go through? Meiosis. So remember, meiosis reduces the number of chromosomes. So from diploid to haploid, the sporophyte has made haploid spores through meiosis. Then the spores will start to divide through mitosis and make the haploid gameophyte. And this cycle continues, okay? So that's really important to know. So I'm going to come back to my plants, and I really want you to see the dominant or the bigger form in each plant that we're going to talk about. So in moss, you see the gameophyte is all the green, and the sporophyte are these tiny little stalks with the capsules at the top. So you can see here the gameophyte is the dominant form for mosses. If you looked at mosses under the microscope, there are particular things that you can see. So if you looked at the gameophyte in a little bit more detail, you can see the difference between the male parts and the female parts. So here's the moss Antheridia, and as you can see, they have these capsules at the top that are the sperm cells, okay? So here is the moss Archaeogenia, and these will produce the egg cells, okay? So these are both the green stuff, the gameophyte, but here's the male part and here's the female part. So going on for ferns, so now I would have showed you a live specimen of a fern. So for a fern, the whole green part is called the sporophyte, and if you look underneath the fern, you can see these little clusters, the sporangia. Okay, so that is where the spores are kept, and this whole structure is the sporophyte. The gameophyte is this really tiny little structure, okay? So now we can see that in the fern, the dominant or the bigger structure is the sporophyte, and the less dominant is the gameophyte. So this is opposite from moss, okay? So if you were to look at this under the microscope, you would have seen, here's the gameophyte. Again, it's really small, and the gameophyte is important for making gametes, right? The egg and the sperm. And then the egg and the sperm would come together and fertilize and start developing into the sporophyte that comes out of the gameophyte. Okay, so again, the gameophyte is this really tiny thing, and the sporophyte will grow out of it, and it will grow into this big fern leaf that you see here. Okay, next are gymnosperms. So gymnosperms are pine trees. So the whole pine tree, the green part of it, is the sporophyte. And actually the gameophyte are the cones. So the cones can be either female or male, okay? So the male will obviously make the male gameophyte, which is the pollen, and the female cone will make the female gameophyte, which is the egg. And actually the egg is in the base of the scale of the cone, okay? And these will come together and fertilize and make the sporophyte gymnosperm. So here, if you were to look under the microscope in a little bit more detail, so here's a female pine cone and a male pine cone. So if you looked in a little bit more detail, what is really interesting in the female pine cone, it's more of those woody pine cones that have those scales, and at the base of each scale is the ovule or the egg, which is the female gamete, okay? And then the male pine cone has these kind of sacs at the ends of them, which contain the pollen. That's the male gamete, okay? And these are more of like the softer cones. So again, these are the gameophytes, which are a lot smaller. So cones are a lot smaller compared to the big dominant sporophyte in gymnosperms, which is the whole pine tree, okay? Lastly, we have our angiosperms. So angiosperms are flowering plants. So the whole flowering plant is the sporophyte. And if you look in this small structure in the middle of the flower, okay, this is where we find our gameophytes. So here's where we will find our egg. And here at the anthers is the pollen or the sperm, which is the male gamete, okay? So this small structure in the middle is the gameophyte. So as you can see in angiosperms, the dominant or the bigger structure is the sporophyte, and the smaller structure is the gameophyte. So here with angiosperms, part of your protocol was to go over the anatomy a little bit more. So the anatomy, obviously, you know, the petals, and then here within the inside, we want to talk a little bit more about the specialized structures. So this whole thing right here in the middle is called a pistil. Okay, at the top is the stigma. It's a sticky kind of like landing pad. Here's the style. The pollen tube comes down the style, okay? This whole thing is called the ovary, and inside the ovary is the ovules, which are the female gameophytes, the eggs, okay? Over here, these long structures coming out, this is called the stamen. It contains the filament, and at the top, the anther. And the anther contains the pollen, or the male gameophyte. And the pollen would come and land on the sticky stigma and fertilize this flower. So you would take your flower, and then you literally would just cut the ovary in half. So here is a screenshot of a YouTube video of them doing a flower dissection. You didn't have to watch the whole thing. I just cut to the chase. So here they cut the ovary in half, and you can see if you look really closely, these like little dots, those are the ovules that are inside the ovary. So the ovules actually will get fertilized and turned into the seed, and then the whole ovary that surrounds the ovules will turn into the fruit. So whenever like you're eating an apple, you're actually eating the ovary of the angiosperm. Okay, so just a really quick recap. I know that was a lot. So again, how did plants evolve? So first the bryophytes, or the moss, evolved into the ferns, the gymnosperms, the angiosperms. In the moss, okay, the gameophyte is more dominant. It's the bigger structure, and the sporophytes are smaller. In the fern, okay, the sporophyte is bigger and the gameophyte is a lot tinier. Gymnosperms, the sporophyte is a lot bigger and the gameophyte is smaller. And the angiosperm, the sporophyte is a lot bigger and the gameophyte is a lot smaller. Okay, so that's the main takeaways that I wanted you to get from that part of your protocol. So let's continue on. So that was part 3 of all the different plants. And the last part is Kingdom Animalia, your animals. So through this protocol, you would have done 2 dissections. You would have dissected the earthworm and you would have dissected the frog. Okay, so it's really important that you read this whole section of Kingdom Animalia so you can get a really good idea of all the anatomy of the earthworm and the frog. Okay, and since we cannot be doing the dissection virtually, I found some really great videos explaining the dissection. So here's the earthworm. So I'm just going to play this. This is from Carolina.
Identify the anterior and posterior end of the earthworm. Notice the anterior end is slightly larger than the posterior end. At the anterior end, locate the mouth and prostomium. At the opposite posterior end, locate the anus. Also notice that the dorsal or top surface of the earthworm is slightly darker than the ventral surface of the earthworm. Now, notice the segments on the earthworm. The earthworm's body is made up of over 100 segments. The enlarged structure beginning at segment 31 is the clitellum. This structure is vital to external fertilization in earthworms. Let's move on to the internal anatomy of the earthworm. Place the earthworm in the dissecting pan, ventral side down. Gently pierce the dorsal wall with the scissors, just posterior to the clitellum. Continue cutting the dorsal body wall until you reach the mouth. Pin the body wall to the pan. You may have to cut away the tissue between the segments to do this. These thin walls separating the segments are called septa. Identify the organs of the digestive system, including the pharynx, crop, gizzard, and intestine.
Okay, so one thing I really want to point out that has to do with your post-lab question are these 2 structures right here that are really important for the digestive tract of the earthworm. So here's the crop on top, then the gizzard. So the crop stores food, and then the gizzard will grind up the food. If you were to push on these structures, the crop is a lot more flexible and can collapse easily, and the gizzard is a lot more hard. And you have to think really carefully, why would the gizzard be more hard than the crop? So remember, the gizzard has to move and grind up the food. So think of what kind of tissue allows the gizzard to move, and that tissue is really thick in the gizzard, giving it its hardness. So that's a key point and a little bit of a hint to help you with that post-lab question.
The circulatory system consists of the 2 blood vessels and 5 pairs of aortic arches. Locate the dorsal blood vessel and the 5 aortic arches. To locate the ventral blood vessel, lift a portion of the intestine posterior to the gizzard. Running along the ventral body wall below the intestine is the ventral nerve cord, a part of the nervous system. At the anterior end of the earthworm, locate the pair of cerebral ganglia, which functions as the earthworm's brain. The ventral nerve cord extends from these ganglia down the body of the earthworm. Locate the excretory organs called nephridia in each segment. These specialized structures remove nitrogenous waste from the earthworm's body.
Okay, and now your next video is from Carolina. It's the dissection of the frog. So here I just sped up a little bit. So the frog was cut through the abdomen. So here you can see some of the frog's skin and the muscular part of the abdomen that the person has cut through. And now we're looking at the internal anatomy.
Look up here where I've cut through the chest plate. I noticed something that appears to be in a bit of a plastic bag, so to speak. This is the pericardial sac, and this is the 3-chambered heart of the frog. We see the 2 atria at the top. I'm going to pull that back. And we see the ventricle at the bottom. This large structure that we see in the center of the frog is the liver. The first thing we want to do when we're investigating the liver, knowing that we may have listed the gallbladder as an accessory organ, or maybe we've listed the liver as an accessory organ of digestion as well. We're going to lift this up and we're going to see this absolutely lovely gallbladder. I'm going to move that somewhat forward so you can see that right here. It's greenish in color. It's a little bit like a small balloon, and I can also see this lovely common bile duct right here. And hopefully your students have added this to their model and they're excited to see this. Also right here, and it's in plain view, and this can be sometimes tough to find, is the pancreas. Looks a little bit like chewed up juicy fruit gum, and it is also leading in right here. These will be important to retain as you're looking at your model. I'm going to remove the liver now so that we can take a better and closer look at this digestive system. So I will just remove these 3 lobes of the liver. On a side note, if you're thinking about really studying the circulatory system of the frog, I would highly suggest that you order an injected frog so that your students can follow the trail of those blood vessels very easily. If I take my probe back up into the esophagus and I follow it down, I will find that it leads to the top of the stomach right here. Here's the stomach of the frog. We notice this shape of it. I'm also going to open it for 2 reasons. It's really fun to see if there are any prey items found in the stomach. Also to look and see if there are any structures that we can investigate within the stomach to talk about their function. Just going to make a small incision here and then cut up this way. And I do notice something that is not at all discernible about what it is or what prey item it might be. But I noticed these ridges or grooves in the stomach. These are called rugae. And this will be a structure that we'll definitely want to ask our students to note in their science notebooks and guess or predict about the function of the rugae. As I follow this down, I'm going to see that lovely pancreas again. And as I come up here, I'm going to notice that this changes in size, diameter, and texture. This is going to be the small intestine. And as I pull this up, I'm going to see that it's coiled and also that it's attached with this type of almost a saran wrap material. This is the mesentery. If I look closely, and especially if I had an injected frog, I would notice that these are blood vessels. I'm going to follow that small intestine down to this spot in the frog where it appears to change in both color and size. This would be the large intestine leading to the cloaca. I would be asking my students why they think that this version of the small intestine is longer, more coiled than this portion of the large intestine. Female frog, we see this large egg mass on both sides, which would be in the ovaries. And then we see these beautiful oviducts that lead to the cloaca and would allow this urogenital opening of the cloaca, which we've removed, to release the eggs into the environment. Here's our male frog. Let's just review a little bit what we're seeing in the male frog that we have seen in the female. Here are the 3 lobes of the liver. What we see, here's the stomach and there's the pancreas. And what we're obviously seeing that was not as visible in the female is we're seeing these fat bodies. When we notice these fat bodies, which were not overly present in the female due to the masses of eggs that were present there, we notice that they're gathered around the reproductive structures. Very much like in human anatomy, these are energy storage features. They're also important in temperature, keeping the frog warm, and also as an energy store during hibernation. I'm going to remove a few of these fat bodies so we can have a closer look later. When looking for the testes in the male frog, it is easy to confuse the spleen and a single testis. The spleen is found in the mesentery between the small intestine and the body wall and is much darker than the testis. The testis sits closer to the dorsal body wall, usually has more of a pinkish coloration, and is located right at the junction of the fat bodies. Also remember to look for symmetry. The testes appear in pairs, but there is only one spleen. Along the dorsal body wall is the dark, elongated kidney. Like the testes, these will be paired. If you're up for a bit of an adventure, you can actually tease this away and find the ureter, which will lead down into this sac right here, and that would be the urinary bladder, and that is present in the female as well. Here are those fat bodies that we removed. We can see their structure as being able to be very flexible and inside the body cavity able to move around and insulate and protect those organs.
Okay, so that was the end of the internal structures of the frog, the dissection. That is pretty much what we do in lab. We try to identify all the internal structures that we can find. So that was a really good demo on what you would have done in lab. So after you have watched that, now you can just go right to your post-lab questions. Okay, so just like before, make sure you have your Word document. You have your name that you're answering your post-lab questions for lab number 11. You can answer them all right here. Number 9 just has 4 parts, A, B, C, and D. Just tell me the answer for all of those. And again, all your answers are throughout the whole lab, so make sure you're really reading that intro and the intros to the different parts of the labs, and that's where you're going to find all your post-lab questions. So again, make sure you save this Word document to your computer, and then you upload it to Blackboard at the submission site. Okay, so this is the end of the lab instructions for lab number 11.